Listen, get note-taking, learning / mode. This is the second half of chapter 10. And I got the first half on video already. Y'all know where to find these videos, right?
No.
YouTube.
What do they call them?
Mr. Willis's awesome biology textbook videos.
Nice.
Just type in Mr. Willis's awesome biology textbook and it'll all come up. You can go to any one you want. So, we got to talk about the movement of particles. Now, I talked about some of this yesterday, but I want to go back over it. So, diffusion is just the random spreading out of molecules. Sometimes, though, to get into a cell, the diffusion has to take place through proteins. We call that facilitated diffusion. Can you say facilitated diffusion?
Facilitated diffusion.
That's when the molecules come through proteins. Now, sometimes they can just go through a hole in a protein. That's called a channel protein, or protein channel. Whoop, just go right through. And sometimes they have to be carried through by a special protein that'll change shape to get the molecule through. We call that a carrier protein. And this protein right here is going to change shape to let the molecule go through. Carrier protein. I might have shown y'all a video of that yesterday. Did y'all see a video of that yesterday?
I don't think so.
No? Okay, I'll show you one in a minute. Now, the normal direction of molecular movement is called the concentration gradient. So I'm going to set some up on the board here. Let's say that this is a membrane right here. You getting this down here? This is a membrane. And let's say we have a whole bunch of particles on one side of the membrane. These dots are molecules. And we got a whole bunch on one side of the membrane. But we don't have much on the other side. Which way do you think these molecules will move across the membrane overall? Do you think they'll go that way, or do you think they'll go that way? 10 points if you can tell me. Will the molecules move to the right or to the left? Where will you get more movement? To the right or to the left? That's all you got to write down, one way or the another. To the right or to the left? Write that down. Make sure your partners agree.
Adam.
Blue correct. Green correct. Orange correct. Purple correct. White correct. Peach correct. Who is peach? You know what?
We were paid.
I'm marking on the wrong sheet.
Oh, no, that's not.
Silliness. Peach. White. Purple.
Am I in that?
Orange. And green. There we go. Yeah, so y'all all got to write this. Moving to the right. These are all going to move to the right because you go from high concentration to lower concentration. We call that diffusion. And we call this direction the normal direction of motion going that way. Do you know what that's called?
Concentration gradient?
Yes, the concentration gradient. The concentration gradient is the normal direction of motion of particles. However, sometimes a cell must move something against the concentration gradient. Cell might need to move these particles that way. To keep all of these here and to move these that way. Cells do this for a lot of reasons. For instance, food. The cell might have a lot of food inside of it, but it wants more food, right? It's greedy. If it sees a food molecule there, it wants to pull that in. So it's got to keep all these inside and pull that in. That requires energy. So that's called active transport. Can you say active transport?
Active transport.
Everyone at the same time loudly?
Active transport.
That's going the opposite direction. That's pulling these things inside, making them go that way in the reverse of the concentration gradient. That requires energy. Let me explain to you an example of this. Imagine, y'all check this out, look up here. Imagine I had a room full of ping pong balls. Imagine it was ping pong balls stacked to the ceiling, because I'm a ping pong ball hoarder, okay? And I'm wading through my ping pong balls to get around the room, and there's a whole bunch of them. What would happen if I opened the door? They go. It would be this massive avalanche of ping pong balls. And if someone was standing out there, they'd be like, oh, and they get crushed by the ping pong balls. It would be a nightmare, wouldn't it? And I'd be upset because I'd lose all my ping pong balls. That wouldn't be good. But let's say the door's closed. So the concentration gradient, if we're thinking about a cell, is going that way, right? It's going out the door. However, let's say I wade through my ping pong balls and I look out the window and I see a ping pong ball out there.
Oh.
I'm a ping pong ball hoarder, y'all. I want that ping pong ball. See what I'm going to do? I'm going to hold all my ping pong balls in, and I'm going to open the door just a little, keep the ping pong balls in, and I'm going to pull that ping pong ball in, and then close the door. And I got another ping pong ball. Wouldn't that be great if I were a ping pong ball hoarder and I could do all that? Did you notice that required a lot of energy to get that one extra ping pong ball in here?
Yep.
That is active transport. Using energy to pull more molecules into a space that already has a lot of molecules in it. That's active transport. And so I have video footage of that, as if that weren't enough. Here's video footage. Could you get the light for me over there, Adam? Video footage of active transport.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. One phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side.
I hate his voice.
Sounds like he has a cold.
So let me do the talking here. So we call this the sodium-potassium pump. It's an active transport mechanism that happens in your nerve cells. Sodiums get pulled to one side, and there's ATP there giving the energy. Did y'all see that? Let me back that up a little bit. So here comes sodiums. Now, they're going to be pulled. This side has a lot of sodiums on it. But these sodiums are going to be pushed into that side. And here comes the little ATP molecule. This is an energy molecule that cells use. So watch this ATP molecule. It clicks there. And then the protein changes shape and lets the sodiums go to the side where there's already a lot of sodiums. And then there's potassiums. These little K's will come in and they'll hook in. And then the protein will change shape. And it'll push potassiums to the side where there's already a lot of potassiums. So this thing is happening in your nerve cells at all times. We call it the sodium-potassium pump. It moves sodiums to one side of the membrane, potassiums to the other side of the membrane. Your nerve cells are made in such a way where they have to have sodiums on one side and potassiums on another for your nerve cells to work. And that takes a constant input of energy. You're just sitting there right now, but this is going on inside your nerve cells. Sodium's being pushed to one side, potassium is being pushed to another, and that allows your nerves to work. We call it active transport. Now, there's other modes of actively transporting materials across a membrane. What we can do is we can take a lot of these molecules and we can pull them inside a cell using what's called a vesicle. Can you say vesicle?
Vesicle.
A vesicle is like a bubble. Imagine if this were a cell membrane. Let me erase these little things. Imagine if this were a membrane here, and the membrane makes a little pocket. The membrane kind of bends inward like that, and collects these molecules, bounce in here, and fill up this space by diffusion. They're just spread out into the space. And then the cell can like pinch it off and close off this bubble and create a bubble inside the cell. Pull in that bubble, and then that bubble can move into the cell. We call that has a name. It's called endocytosis. Endocytosis. Can you say endocytosis?
Endocytosis.
Look at y'all saying it loud the first time. I love that. Endocytosis is pulling in a bubble full of material. And that's what you see. They're trying to draw it there for you. There you are. This bubble here, see this material this area being pinched off and pulling in material. Now, if the molecules that it's pulling in are really tiny, it's called Pinocytosis. If the molecules that it's pulling in are really big, it's called Phagocytosis. Pinocytosis if it's pulling in small molecules, phagocytosis if it's pulling in big molecules. When I was a kid, this was called Pinocytosis and Phagocytosis. And since then, they've changed the name to Pinocytosis and Phagocytosis. I think it's because it sounds dirtier if it's Pinocytosis and Phagocytosis. But I don't really know why. It just has. I remember in college kind of laughing, going Pinocytosis. That was kind of funny. And now that's just they call it Pinocytosis or Pinocytosis. So I don't know why they changed it. But anyway, that's taking in molecules with these vesicles. And you can also release molecules with vesicles. If you see this big vesicle here that contains like a bubble in the cell, can you tell it's made of membrane? Do you see that's membrane?
Yeah.
Can you tell those little tiny phospholipids? There's a bunch of them. And they make this membrane around all the particles, and then they go to the edge of the membrane and they squirt out a bunch of particles. We call that exocytosis. It's the opposite of endocytosis. Endocytosis is pulling in a vesicle, and exocytosis is excreting a vesicle. Exocytosis. So the cell can do both. It can pull in vesicles or it can spit out vesicles.
What happens to all the extra lipids?
Well, they actually become part of the membrane here. So these lipids there will just become part of the membrane, and the membrane will be a little bit bigger. But at the same time that this is releasing, you could have another vesicle somewhere else forming, pulling stuff in, and so the membrane would stay about the same size.
Is this what causes cells to grow?
Yes, you can cause cells to grow by adding more vesicles to the membrane. Yep. And the cell will do that. The cell will make more phospholipids and send them to the membrane, and they'll merge with the membrane. And that's how cells can grow. Do it, Sam. That's good thinking. You want to see video footage of this?
Yes.
On the top.
I got lots of videos showing this. This is good stuff. Here we go. Enable all. Not recommended. I'm going to do it anyway. I'm living dangerously, y'all. Uh-oh.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer of the plasma membrane.
What is going on?
They leave the cell.
The endomembrane system consists of the
Hold
endocytic
on.
reticulum.
Too
and
dangerous.
Golgi
Oh, here we
interactions.
go.
Vesicles pass from one.
Okay. Sorry, I shouldn't have done that. I lived dangerously and then I paid for it. Try this again. Yeah, here we go.
This is a cell that secretes large molecules that are unable to pass through the lipid bilayer
Great
of the
video.
plasma
Watch
membrane.
this.
They leave the cell instead by a process known as exocytosis. After production in a system of membranes called the endoplasmic reticulum, the molecules are packaged in a small sac of membrane called a vesicle.
Here's a
The
vesicle.
vesicle moves to a stack of membranes called the Golgi apparatus.
We'll learn about that
The
later.
membranes merge, and the vesicle releases its contents for modification. As the molecules leave the Golgi apparatus, they are again packaged in a vesicle, which moves to the plasma membrane. As membranes merge again, the contents leave the cell without actually crossing the plasma membrane.
Whoop. How cool is that? Exocytosis.
So,
This process can also occur in reverse, allowing large molecules to enter a cell. This is termed endocytosis.
Huh? Did you like that?
Some good video footage.
Wasn't that good? Let's see it here again. So here comes the vesicle.
so the small molecules can just filter out, diffuse out.
Yeah, they diffuse out after they're released.
If they're not large.
So these things will diffuse away. Phew. And if we go backwards, these molecules get pulled in, and the vesicle breaks off, and now they're in a vesicle inside the cell. Now the cell might digest those pieces, it might just keep them in that vesicle. Sometimes it hits them with special organelles that destroy them. Who knows? Depends on the cell. What did you think about that?
That one was very good visual.
Yeah, I agree. I got more of them. Here's another one.
The endomembrane system consists of the endoplasmic reticulum and Golgi apparatuses. Vesicles pass from one part of the endomembrane system to another, and secretory vesicles from the Golgi apparatus merge with the plasma membrane,
Look at this vesicle.
secreting their contents by exocytosis.
Watch it. Isn't that good?
Protists, such as paramecium, sweep food particles into their gullet, trapping the particles in a food vacuole. Digestion takes place within the food vacuole, and the products of digestion pass through the membrane of the food vacuole. Finally, the food vacuole merges with the plasma membrane, expelling waste products into the environment.
That's paramecium poop. Here's phagocytosis.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement.
Okay, so this is like a white blood cell. This is what your white blood cells do for your body. That's a bacterium. That thing could harm the body. If it starts multiplying inside the body, you could get an infection. Have you ever heard of an infection?
Mm-hmm.
Yes, sir.
So that thing needs to be destroyed. Just one of those can kill you if it multiplies enough and forms lots of them. So this thing will crawl up to it, it'll grab it, then it'll pull it inside by endocytosis. Yeah, that's it. Surround that thing. And now it's in there, and now the cell's going to hit it with a bunch of organelles called lysosomes. Lysosomes have digestive enzymes that'll kill it. They hit it with some lysosomes, there we go, and that's going to break it apart. Kill that thing. We want that thing dead. Yeah. Here we go. Oh, here we go. Tears it apart, destroys it, and then it's going to release the contents out of the cell
And it's
by
not bacteria.
exocytosis.
So then where did those go?
Those just float around outside the cell. What will end up happening is they'll go to your kid. They'll get in the bloodstream. Your kidneys will filter them out of the bloodstream, and you'll pee them out. That's part of the waste that comes out of your body is pee. Stuff. This thing's killer.
Oh, I wonder if that's all.
Literally.
Phagocytosis, which means cell eating, is a form of endocytosis. A cell such as a phagocyte engulfs surrounding material, forming vesicles called phagosomes, derived from the plasma membrane. Phagosomes merge with vesicles called primary lysosomes that contain digestive enzymes, forming secondary lysosomes. Digestion takes place within the secondary lysosomes.
Killed it. That one is good.
So phagocytosis is for bigger molecules.
Bigger molecules, yeah. Big molecules being broken down, that's or being taken in, that's phagocytosis. And then pinocytosis is smaller molecules.
Pinocytosis, which means cell drinking, is a form of endocytosis. A cell brings in droplets of the environment by pinching them off with the plasma membrane. The pinocytic vesicles break away from the membrane and move to the cell's interior.
So these things each contain liquid or water or something inside them. And so the cell can use that to bring in water.
Just for pinocytosis.
That's pinocytosis, yeah.
What about air?
So phagocytosis.
It could bring in air, possibly. What were we going to say?
So phagocytosis is eating and pinocytosis is drinking?
Yeah, basically. Yeah.
Cells also use energy to transfer materials in bulk,
This is pretty
but
good.
this time by forming membranous sacs that hold their contents under wraps. In a process called endocytosis, part of the surface membrane encloses the material, forming a sac which brings the contents into the cell's interior. In the opposite process, called exocytosis, the sac moves through the cytoplasm to the membrane, fuses with it, then releases the contents.
Huh. Isn't that cool?
Yeah.
That had cool music, too. All right, y'all pretty good? Y'all pretty good with this?
Yes, sir.
Okay, you can stop filming. I got a few more questions.